Good evening, everybody. For those of you who don't know me, I'm Mark Roithmare, and I have quite the privilege to serve as the CEO of the Alzheimer's Drug Discovery Foundation. And I want to welcome you all to the fourth in our scientific symposium lecture series here at the ADDF here in Palm Beach. Lots to kind of celebrate tonight. For all of you who were with us for the gala that was at the beginning of March, the event raised over $1 million, which was wonderful. And we have all three of our chairs from that event that are here. Ms. Nancy Goods and Ms. Bonnie Lautenberg, yes. And also Heidi McWilliams, and I'm going to brag on Heidi a little bit more in a couple of seconds. We have entered into, just in the last 3 years, what Dr. Phillip and I were recently in London and was being called by the World Dementia Council a new era for Alzheimer's. And this era is distinguished by, in the last 3 years, several things. Number one, we now know there are things you can do to help prevent dementia and Alzheimer's. Tactical things that you can do to push out cognitive fuzziness, right? So terrific. We also know now that there's a test you can take that can tell you if you are going to be susceptible to Alzheimer's. And on top of that, part of what you're going to hear tonight, which is further exciting news, is that not only can you do something about it through prevention, but we're now starting to see some of the first therapies that are coming about that can actually treat. Okay, and you're going to hear about a lot of phase 2 clinical trials that we're doing now, that we know it's safe and it's starting to show some hope. And as Leonard Lauder said a couple of years ago, he said there was hope on the horizon for Alzheimer's, and now Leonard says the hope's knocking at our door. And you're going to hear about that today, so which is terrific. In this series, we've had some phenomenal doctors present, Dr. Robbie Britton, Dr. Frank Longo, Dr. Mia Kivipolto, and just laying out the gamut of what this new era is about. Dr. Cummings tonight will be no less impressive and informative as we go. I want to come back to Heidi McWilliams and Tom McWilliams. Tom's on the board of the Alzheimer's Drug Discovery Foundation, as is Larry Leeds, who's here with us tonight. But Tom and Heidi, about 2-3 years ago, came to the Lauder family and came to the ADDF and said, we need a scientific symposium lecture series in Palm Beach, that ADDF is only about one thing, the science, and we need to bring that science out and show it to all. What's been terrific in this series, literally, some of the top doctors, not just in the United States, but in the world, have been part of this series. So Heidi and Tom, we really, really thank you for your vision on this. And this being our final of this year, thank you for everything that you've brought to us. It's my pleasure at this time to turn the proceedings over to Dr. Howard Phillip. You're going to hear about some really, really interesting science tonight at the end of the presentation between our two doctors. There's going to be opportunity for Q&A. And then after Q&A, there's going to be some refreshments for everybody. So we have a lovely, lovely next hour or so that's in front of us here. Dr. Howard Phillip, he has been doing this for over 40 years, but he's very young, right? He started doing this at 7, so it's just terrific. Dr. Phillip has worked in academia at Rockefeller University in New York City. He has worked in industry. 24 years ago, Leonard and Ronald Lauder came to him and said they had an idea to start a foundation called the Alzheimer's Drug Discovery Foundation, and that the foundation would focus on only one thing, translational science. How do you take good ideas in the lab and get those good ideas commercialized and into patients? And the Lauders were willing to put up all of the underwriting to the foundation so that we could say to every donor, every nickel you give us goes directly into the clinical trials and the prevention and the search for biomarkers that we fund. And the last piece of it is that the ADDF would not be giving out grants per se, it would be making investments, so that if there was an academic institution we were going to put money into, That went as a contracted investment to them so that if that idea materialized into anything, there would be a portion of the proceeds from that that would come back to ADDF that we'd put in the science. Similarly with biotechs, that if we would invest in them, someday if they saw what they call deal flow, we'd see that and the money go back into the science. And this is a very niche thing. It's called venture philanthropy. 24 years ago, with the exception of maybe cystic fibrosis, there were not people talking about venture philanthropy, but we're here 24 years later. I give you all that as background because they mentioned this all to Howard 24 years ago, and they said, Howard, what do you think of this idea? He said, what a great idea. You're exactly what you should be putting your philanthropy into. He wasn't prepared. I don't know if it was that day or the next day when they came back and said, Howard, who, by the way, Howard's a physician and a scientist. They said, Howard, how about you give up your day job, you can keep your small private practice, and you be employee number 1 at the Alzheimer's Drug Discovery Foundation. So it is my pleasure at this point to turn it over to the co-founder of the ADDF and our chief science officer, the man who is our true north, Dr. Howard Phillip. Howard?
Thank you, Mark. I want to thank all of you for coming today and joining us in this effort to conquer Alzheimer's disease. I just wanted to say a few words, and then I'll introduce Dr. Cummings. We actually have a scientific strategy. I'm a geriatrician. I've been specialized in taking care of older people and people with Alzheimer's disease for over 40 years. But being a geriatrician has given me a kind of unique perspective over this time, in the sense that if you think about what the leading risk factor for Alzheimer's disease is, it's aging. And one of the problems in the field has been that there's been, in my view, such a strong focus on 2 pathological hallmarks, the amyloid plaques and the tau, but not enough on all the other factors that contribute to aging of the brain and other organs, such as inflammation, genetic changes, metabolic changes that are related to diseases like diabetes, vascular changes and a risk factor of hypertension for cognitive decline with old age. All of these what we call medical comorbidities. And so that's what we've been doing all these years, is funding innovative ideas that basically are not exactly what Big Pharma is doing and what you've heard about in the last year or so. We've moved a lot of our portfolio into clinical trials. And to conduct good clinical trials, Mark mentioned the blood test, and also there's a brain scan on the market. And we're very proud that from the very beginning, back in 1999, we started funding the development of a brain scan, a PET brain scan. PET is the kind of brain scan you can get at your local radiologist to detect the plaques in the brain that Alzheimer's saw many years ago in 1906. And just to fast forward, that test was the first diagnostic test approved by the FDA for evaluating patients and making a diagnosis in practice of Alzheimer's disease. But I think even more importantly, the reason we helped to develop that test was to accelerate drug development. Because if you think about it, imagine trying to develop a statin drug for heart disease and you wouldn't know how to measure cholesterol. So cholesterol is a biomarker for heart disease. And in Alzheimer's disease, we now have these biomarkers, and Jeff and I will talk about that. But one of the major advances we've had is not just the fact that there's a brain scan that I can order for someone. They go down the road to a radiology office, and within 24 hours, I can tell that person with about 90% certainty whether or not they have Alzheimer's disease. Of course, the brain scan is a little expensive, but there's a blood test now that's been approved and is on the market. Another company that we invested in called C2N, which was spun out of Washington University. And through our program that we have, about $100 million program that we have with Bill Gates and Jeff Bezos and Mackenzie Scott and David Dolby and people like that, we're developing other biomarkers, not only to improve clinical diagnosis, but more importantly, I think, to accelerate the development of new drugs, because we have to have these biomarkers. With that really brief introduction, I want to say that the reason Mark said we're in a modern era is really because we have these biomarkers that can help us accelerate the development of new drugs. Last thing I'll say is that Dr. Cummings puts out every year a survey, which he can tell us about, of all the new drugs that are being developed. I just heard him say there's 147, and he found that 72% of those drugs being developed now are for the non-pharma type of targets, the beta amyloid targets, the tau targets, that big pharma is attacking, and going after these targets that are related to the biology of aging. So I feel that the diversity in targets that's being conducted now really is a validation of this strategy of understanding how aging and the aging brain ultimately leads to Alzheimer's disease. Dr. Cummings, and if I can call you Jeff here in this, you know, I have to say, I've known Jeff for a long time. He and I started together unbeknownst to each other back about 40 years ago. But Jeff really and truly is one of the world leaders in Alzheimer's research, and particularly in clinical trials. He's known all over the world. Jeff is incredibly prolific. He's written over 800 scientific papers that have been published and 44 books. And we just did a book together, which Jeff can tell you about. He's an endowed professor at the University of Nevada in Las Vegas and heads up the Alzheimer's Center and the Center for Transformative Neuroscience there. He is a professor at the Cleveland Clinic as well, and was formerly the director of the Alzheimer's Clinic and program at UCLA before moving to Las Vegas. He also consults with a large number of biotech companies and really understands drug development, clinical drug development and clinical trials, I think, more than probably almost any person in the world, and I don't think that's an exaggeration. And with that, I'll just start off our dialogue here by asking Jeff about his perspective on how clinical trials have changed and why we think we're kind of in a new era where our success rates might improve compared to what they have been over the last 20, 30 years.
Well, thank you, Howard, and thanks for coming this afternoon. What a great thing to have a crowd here, and here it is Tuesday afternoon, and you're all out on the lawn. This is great. This is what we love, and of course, Howard and I love to engage people and answer your questions. We're a little bit more interested in answering your questions than we are in the dialogue here, because you're here to get answers, and we don't know what questions you have right now. But we'll have a dialogue for a while, and I'll start with clinical trials because I do love clinical trials. It's a very special way of getting information. So what's a clinical trial? It's a very structured way of testing whether a drug is working, and it's almost always compared to a placebo. So people have a lot of concerns about that, right? Well, my mom might be on a placebo. Well, she only has really one chance of being on the drug, and that is to be in the trial. And nearly all of our trials provide the drug absolutely with certainty when the blinded period, the placebo period, is over. Because we have to learn that, because that is the only kind of information that the FDA accepts. So no placebo, no drug. So we have to do this. And it's very important because the placebo is often the most informative arm of the trial. You must see the placebo going down to know that your drug is keeping the patient well. So that's the only way that we can tell that. And we have made so much progress in terms of clinical trials. The design of clinical trials has been mathematically much more sophisticated now than it ever was before. And as Howard mentioned, the biomarkers. A biomarker is a way of measuring something that your body is producing or something that changes in your body as a result of giving the drug. So let's say that you have an abnormal protein in your blood. We give a drug and see whether that protein goes down. Or we look at the brain scan and we give a drug and we see whether compared to placebo, there's greater removal, for example, of the protein. And these tools have become available to us for the very first time. Howard, you made a wonderful example of how could we treat heart disease if we couldn't measure cholesterol in the blood, right? How could we have done that? Well, that's what we've been doing for the last 20 years or 30 years, is trying to develop drugs without having biomarkers. And these biomarkers have transformed our lives. They're the new toolkit for measuring disease in people and for measuring the response to treatment as we test new drugs. There's no question they're going to accelerate drug development. I'll stop there.
No, thanks, Jeff. So in terms of your view of what's going on now with the 147 trials, not counting the amyloid trials that we could talk about later, what are you most excited about in terms of new targets and new drug classes that you see on the horizon might be useful for patients with Alzheimer's disease? What's in the pipeline, basically?
Thank you for that. As Howard mentioned, we write an annual review where we look at all the drugs that are in the pipeline. And there's 147 trials of about 135 drugs right now in the pipeline. That compares to about 3,000 drugs if you were to look at cancer right now. So you can still see how far we have to go. Because if you're getting, I don't know, 2% approval of drugs, then 2% of 3,000 is a lot of drugs. And 2% of 150 is only a few drugs. So we really need to do much more of what ADDF is doing, to invest in trials, because we have to have more trials to discover more drugs, no question about it. Some of the most exciting things, we have learned so much about inflammation in the brain in recent years. And as we learn about the biology of the brain, that's also how we learn about new targets that we can develop drugs to. So if you get an injury to your knee, it swells up because it's inflamed. Well, you can imagine what's happening in your brain when there's inflammation in the brain. And now we can see that. We can even see it with scans. So some of the drugs are pointed at inflammation. There's a very interesting antibody, for example, that's specifically addressing one aspect of the inflammatory cascade called TREM2. So I think that's a really interesting drug. There's a drug where we're looking specifically at whether this overlaps a little bit with tau. But there's a drug that's in the pipeline, pharmatropic. Some of you know this drug, and it has multiple effects. We think a lot about combination therapy. Could we give more than one drug? And I think we're going to have to. But another way of thinking about that is, could we have one drug that has multiple effects? I think the pharmatropic drugs looks a lot like that to me. So I like that drug a lot. The synaptic drugs, the connections between nerve cells are synapses. They're the connections. And they're the first thing to come apart in Alzheimer's disease. The very first thing. That's the first measurable thing we can see. So what about, could we strengthen the synapse? So there's several drugs, including cognition therapeutics that ADDF has worked with, that is specifically looking at preserving the synapse. So there's a lot of excitement about different categories. One of the drugs I'm working with personally is called semaglutide. You might have heard about this because it just was approved for the treatment of obesity in the United States. It's been approved for the treatment of diabetes for quite a long time. And one of the really interesting things about Alzheimer's disease is there is a problem with sugar utilization. Some people call it the third type of diabetes. And also diabetes is associated with inflammation again. And this drug, this semaglutide, reduces inflammation as well as increasing the access of glucose to the brain, or the sugar of the brain.
So thanks, Jeff. I just want to say that in terms of semaglutide, which Jeff mentioned, which is, if you see it on TV on the nightly news, it's called Rybelsus, I think, Rybelsus. So that's a drug for diabetes that's been being repurposed for Alzheimer's disease. So repurposing means that you take a drug that's on the market for one disease and you test it in another disease when there's a rational reason to do that. And ADDF has had a very large portfolio of repurposed drugs. And in fact, the one of the contributing factors to this very large semaglutide study that Novo Nordisk is doing is that ADDF funded the Imperial College of London for a related drug called liraglutide by Dr. Paul Edison there several years ago, which had some promising results. And so that's kind of what ADDF does, is we do run these small, what are called phase 2A exploratory studies to see if there's a signal there, and then let the big guns come in. So for $3, $4, $5 million, we can look at a drug and see if there's any kind of reason to think that there should be a bigger trial. And then the pharma companies or the NIH can come in and bring in $25, $50, $100 million and run a much larger study to see if that signal is really true. Jeff also mentioned a company called Pharmatropics that we're really excited about. And this is a company that's developing drugs that are what we call neuroprotective. So if you think about all the factors that lead to aging, there's multiple factors. There's multiple ways that a brain cell can get hurt and die. As Jeff mentioned, inflammation and so on. Imagine if there was a drug that could simply be neuroprotective regardless of the course of injury. It could stimulate the protective pathways in the neuron to help it survive. And we started funding this program more than 20 years ago when a doctor named Frank Longo was at UCSF. And then he moved to become chairman of neurology at UNC, and we continued to fund him. And during that time, about 2005, we encouraged him to spin out a biotech company. We were among the founding investors of that biotech company. Frank then moved to Stanford, where he's currently the chairman of neurology and neuroscience, and continued his work on developing this drug. We continued investing in that company. And to take that from the very earliest stages of drug development in 2000, where he screened on a computer screen 2 million molecules against this receptor that was thought to be the neuroprotective receptor. It took a year to do that. Today you could do it in about a week on 2 billion compounds. But he found out of that 2 million compounds, 60 that seemed to fit in the pocket. And from that, he bought the 60 compounds in powder form. He tested them on cells to see if they were neuroprotective. Out of that, he found 2 compounds that protected brain cells in test tubes from being injured by amyloid and other mechanisms. And those 2 compounds eventually went into clinical development. And fast forward today, he's in phase 2 clinical development. So that trial in phase 1 might cost $5, $10 million. The phase 2 trial probably costs somewhere around $15 million. But now the big step up is going from about 150, 200 patients to like 5, 6, 7, 800 patients, maybe 1,000 patients. And the step up in costs goes to 50, 100, 150, $200 million. So now, in the course of the biotech development, this company called Pharmatropics, he needs to raise money from venture capital or from really large grants from the NIH, which is a lot of money now. And so that's the challenge now. The barrier now is not scientific. He's been able to show that there's a really potentially revolutionary drug, which could be applied in many neurodegenerative diseases, not just Alzheimer's, did work on a multitude of biomarkers in a phase 2A study, showed that it really changed the body's response to the drug in terms of inflammation and other things. But now he's got to get really large numbers of people to show that the drug actually slows down the clinical disease, because Jeff can talk more about this, but to show that the drug actually slows down the disease takes a lot of patience because there's a lot of variability. And so the challenge is really in drug development. And once we get into phase 2B and phase 3, now we're talking about $300-400 million per study. And the only entity that can really do that is industry. So we have to have this ecosystem of development, venture philanthropy in the earliest days of risk, working its way through to early investors in biotech companies, NIH money, and then finally, as it gets into the real challenges of late-stage clinical development, the really large amount of dollars that's required. We've been following Frank for over 20 years. That's how it takes. So we're talking 12 to 15, sometimes even longer, hundreds and hundreds of millions of dollars. And the failure rate is over 90%. And I'm not talking about our failure rate, which is probably even higher than that because we're taking risk. But it's said that the average person who works in the pharmaceutical industry, and this is from a person named Judy Lewent, who was the chief financial officer at Merck for many years, that the average person who works in the pharmaceutical industry will spend their entire careers never working on a drug that comes to market. And that's because it's so hard. Imagine we're going to have a little white pill that's going to prevent and treat Alzheimer's, and it's going to happen. I know it's going to happen in the next coming years because now we have finally these biomarkers. We can measure the effect of drugs in your blood, and this is really incredible. So we really are in a modern era. I'll stop now, but I just want to say I ran a clinical trial of estrogen in postmenopausal women back in the early 1980s when I opened the Alzheimer's clinic at Rockefeller University in New York. And when I look back on what I did back then, I think I had no idea what I was doing. We were really shooting in the dark. But now, we really know much more about what we're doing. Don't you think, Jeff?
I do. And I want to pick up on Howard's repurposing theme, because repurposing has a great advantage that the ordinary drug goes through phase 1, where you see if it's safe, and then into the patient population at phase 2, and then a big trial in phase 3, and then it goes to the FDA. If a drug is repurposed, you don't have to do phase 1 because it's already been tested in people, the safety is already known, the dosing is already known, complex things like the distribution channels are already established. So there's a whole framework to step into with a repurposed drug. And that's what we did with rasagiline, which was a drug I was really interested in, and ADDF funded that trial for me. And it was fantastic because this was a drug which was already approved for Parkinson's disease. And in the trials where it was approved for the motor improvement, that is, the people could walk better, when we looked at the neuropsychological data, we could see that their memory was improved. So we thought, well, we should capitalize on that observation. So we proposed a trial, and Howard and ADDF helped me propel that trial into the forefront of how trials should be done. Because exactly as you've said, we used a biomarker to identify the people. We used a biomarker as the outcome. That was the brain working better. We did the first clinical trial in which the neurofibrillary tangle scan was actually used in a trial. And we worked with a scientist that had specifically been funded earlier to train, to develop measures on brain scans. So there were so many things that ADDF brought to that trial, and we showed that the drug worked, that there was less decline in the metabolism of the brain in the treated patients versus the non-treated patients. We showed that there was a difference in the accumulation of tau, and we showed that there was a difference in the quality of life of people who responded to the drug versus those who received the drug versus those who received the placebo. So those are all learning things, and sometimes you can't get a drug from phase 2 to phase 3, but you can always learn from that trial. And that's what we're doing right now. We're going back and looking at that blood. We're looking at the relationship of these new blood tests to the scans we did in the trial. So we'll see what happens to that, but we learned an enormous amount, and that can help all other drugs in the that are in trials. That's why it's so important.
And I think what's exciting as a scientist, as a clinical scientist, is that what we're talking about is human research. You know, we've cured mice with Alzheimer's disease about 700 times. But we just had our first drug approval a few months ago from Biogen in 20 years. We've had a 100% failure rate in our disease. But I believe that's going to change. The reason is Alzheimer's, among many diseases, Alzheimer's is uniquely a human disease. It's a disease of the human mind. You know, mice don't write books. And there's a level of cognition that's affected in Alzheimer's. Think about it. Alzheimer's disease affects your ability to think. The highest order of human endeavor, what we call abstract reasoning, is impaired in Alzheimer's. Think about the biology of that and how Alzheimer's affects abstract reasoning. And now we want to treat that to improve it. And we actually funded a repurposing drug called rotigotine, which is another Parkinson's disease drug. But in Parkinson's disease, there was reason to, with this drug, which affected what's called the dopaminergic pathways, there was reason to think that that drug would improve abstract reasoning and other related higher executive functions. So we ran a trial in Rome, Italy, of rotigotine, and sure enough, it improved what's called executive function, abstract reasoning, multitasking, judgment, and so on, the highest order of human thought. So now we're looking at the next steps. So Jeff mentioned repurposing drugs and the advantages of it, that you can get to market more quickly because you don't have to do all that animal work. But there are challenges, commercial challenges, in getting these drugs to market. And I think the next phase of our work at ADDF, which we're doing actually in close contact with our board and with Tom McWilliams in particular, is figuring out ways that we don't want these papers, these studies like Jeff's rasagiline paper or the paper that was published out of Rome, to end up papers in the medical literature that are then buried and never used again. We want to make sure that the drugs that we're studying that are repurposed eventually come to market, because we need these repurposed drugs to come to market so they can be marketed and create what we call patient access. Let me give you an example of that. Thalidomide, I mean, there's many examples, but thalidomide was a really bad drug in the 1950s for pregnant women, caused all kinds of problems in the fetus and so on. But somebody had the idea that thalidomide could work in multiple myeloma, totally different disease. And they developed the drug, and eventually that drug was sold, thalidomide essentially, as a drug for multiple myeloma. The company that did that at the time was a startup biotech called Celgene. And for those of you who are investors, it was one of the most successful biotech companies of all time from an investor point of view. And it's an example of how a repurposed drug can be commercialized and then get to market. Otherwise, we can't advertise it, we can't let people know, doctors aren't going to prescribe it. There's another drug that I would talk about briefly called riluzole. Now, we were talking about this last night. Among all the neurodegenerative diseases, there's never really been shown any drug, talking about ALS, Parkinson's disease, Alzheimer's, that slows it down, except for one or two in the context of ALS, and one of them is called riluzole. And I won't go into the chemistry of how this works, but we tested it in New York and in a clinical trial, and it really seemed to work. And I have to say that I, in my small practice, have been using this drug, and in 40 years of practice, I've never had a patient or their loved one, their spouse, come in and say, that riluzole is amazing. And that's a quote. Another wife of a patient came in after we put him on riluzole and said, the response to riluzole was stunning. Now, I take that as a scientist as an anecdotal report. That's not scientific proof. But when you hear somebody say there's improvement, that gives us kind of the more ammunition, and we know the drug is safe, to bring riluzole to the next step. And that's what we're thinking about. How are we going to get a strategy to bring riluzole to the next step so we can have commercialization? And we're looking at things like patents, novel patents, which are the driver of commercial development. That's part of what ADDF is doing now. I thought another thing that's exciting, to change the topic a little bit, is when we talk about biomarkers, we're mostly talking about brain scans, which we've had a real revolution in brain scans through MRIs and being able to use the data from MRIs in computerized analyses that Jeff and I use in our practice. So it's not just a radiologist kind of looking at a scan and saying, oh, yeah, the brain is shrunken. There's no doubt about it. But now we can run the data through a computerized analysis and say that brain is shrunken by 44% compared to normal people of the age. That's really useful. These PET scans that I mentioned, We have spinal tap information. We have blood tests, incredible progress in blood tests. So you're going to see a whole bunch of blood tests coming to market for Alzheimer's. If you asked me 5 years ago if there'd ever be a blood test for Alzheimer's, I would have been very skeptical. There really aren't any blood tests for neurodegenerative diseases on the market. Alzheimer's is the first. And there are now digital biomarkers, which means that we can do cognitive testing on your smartphone. We can measure your pace of gait on your smartphone. We can arm your house with digital biomarkers. We can detect your speech on your smartphone or your phone and detect very early, before there's even any symptoms that are measurable, before the wife comes in and says, my husband has memory problems, we can detect changes in speech that are indicative of Alzheimer's disease 10 years before there's even symptoms. And working with the Gates Ventures, we have a very large program going on where we're developing what's called a speech consortium, where we're going to be going around the world and developing databases so that scientists can analyze speech trends in patients. Which leads me to one more comment, and I'm going to turn it over to you, Jeff, which is another major advance that I'm really excited about is using these biomarkers, using the brain scans and the blood tests and the spinal fluid tests. We can find people with Alzheimer's disease 20 years before they get symptoms. We know now that Alzheimer's starts not when you're 75 and you're losing your memory, but when you're 50 and you just start accumulating this pathology in your brain. The same way that with cholesterol, you start developing plaques in your heart when you're 30, 40, might not have the first heart attack till you're 70 or 80. That's what's going on now with Alzheimer's. And there are clinical trials, prevention trials, using all this new technology to see if we can detect people in the community that have Alzheimer's disease in their brain but have no symptoms from it yet, and then treat them to prevent them from getting Alzheimer's disease. And if you want to comment on some of those preventions, you're probably much more involved in that than I am.
Thanks, Howard. I was thinking about so many things as Howard was going through those comments. I think it is worth talking about the blood test a little bit more. There is an approved blood test for Alzheimer's disease. You can go to your doctor and you can ask for it. But like all learnings, it's a little bit tricky because Howard mentioned that we know that the protein of Alzheimer's is in the brain for about 20 years before the person gets symptoms. So when we do the blood test, it reflects very precisely whether that protein is in the brain. Gives us a lot of information. But we don't know whether you're 20 years away or 1 year away from becoming symptomatic. So what's really exciting is that there's a second blood test, which when it changes, shows that you're very close to stepping into the phase where symptoms will occur. So it's actually going to be a combination of these two tests. One is the protein in the brain? We can see that in the blood. The other is how close are symptoms? And of course, that's what's really important. But we want all of this because now we can do prevention trials. We're already doing them. There are 6 prevention trials going on right now where people have to be normal to come into the trial. But we have to do a scan and show that they have the protein in the brain. So it's there. It hasn't become symptomatic yet. And the idea is treat patients so that they either never get Alzheimer's disease or get it much later than they ordinarily would. How great is that? We're thinking about preventing Alzheimer's disease. I mean, we would never have been able to think about that just a few years ago. And we can do it because of the blood tests and the way the blood tests are now coming together to inform us about what's going on in the brain. Why has this happened? Because of the advances in the technology. Ultra-sensitive measures have been developed for blood tests. And we can just see things that we could never see before. That's why we didn't believe we'd ever have these tests, because we didn't have the technology to measure it. Now with the technology, we can measure it in a dramatic way. But I want to say another thing, Howard, which is Howard and ADDF developed phase 2 trials In fact, they led a task force, I'll call it, to develop rules for phase 2 trials, because they're different than when you do large trials. And that paper has proven to be enormously influential in the field. Phase 2 trials are, of course, how we decide whether a very expensive phase 3 trial should be done. And much of the world, including biotech companies and big pharma and many other people, do not understand how this phase 2, phase 3 business works. So we just put together a book called Alzheimer's Disease Drug Development, a Research and Development Ecosystem. And we're trying to really understand how the different parts work together with each other, and can we make each part work better than it currently does? Can we make phase 2 articulate better with phase 3? Is that something we could take on? Well, we can't take it on until we define it. So the book is something I'm really happy about, because we're starting to define what the ecosystem looks like.
And I just want to change the conversation a little bit from the research and the clinical trials to how clinical care is going to change. I remember when I started out, when I went to medical school, Alzheimer's disease was not in my textbooks. That was in the early 70s. When I trained in New York and taking care of old people in the hospitals as an internist, I never even thought of making the diagnosis of Alzheimer's disease. When I embarked on my research career and opened the clinic at Rockefeller in 1981, people said to me, "What do you do?" And I said, "Well, I'm doing Alzheimer's research and taking care of Alzheimer's patients." And they would say to me, "Old-timers disease? I never heard of old-timers disease." Doctors that are practicing today, don't have Alzheimer's disease as part of the mainstream of their care. And part of the reason is that you need to do cognitive testing. And most primary care doctors don't have the time, they're not paid, and they're not trained to do cognitive testing. And that's a very important first step in making a diagnosis and getting people into the diagnostic pathway. Once we have blood tests, I believe that Alzheimer's disease enters into the mainstream of the usual workflow of a primary care physician. And I can tell you in the United States that 70% of prescriptions for Alzheimer's disease are written by primary care doctors. And our managed care organizations don't want a lot of referrals unless they're complex cases. That's not true in European countries. So what we want to do is enhance clinical care. And it's also important, I think, so this blood test, I think, will really enhance the way we provide care for Alzheimer's. It'll help doctors enter into the mainstream of their workflow of everyday practice. Somebody comes in with memory problems, maybe they'll do a cognitive test, but very likely they'll be happy to order a couple of blood tests to see if you have it or not. The other thing is that less than 1% of people with Alzheimer's disease in the community go into clinical trials. And if we don't have clinical trials going on, we cannot discover new drugs. And those $400 million studies that are in phase 3, why is it so expensive? Because $200 million out of that $400 million is spent on finding and enrolling the patients. To enroll one patient into a clinical trial, you basically, and again, I'll look to Jeff because he's much more of an expert on this than I am, you have to evaluate about 10 people to find one person who qualifies for a clinical trial. So $200 million. But imagine if instead of having to do an $8,000 or a $5,000 brain scan, which we have to do now to make sure we're getting the right patients into the trials, which is critically important, imagine a blood test that costs $1,000, and even the price of that will come down. And that's why the blood test is not only so important for clinical care, but it's important for advancing research.
So, good doctors, first of all, how terrific are they? Who's more enthusiastic than the other? Really fabulous. Jeff, I want you to hold that thought. Don't forget it, okay? We want to open this up to questions because we know folks have questions. So, and we have some microphones we're going to get around. So we're going to start with Bonnie, and we're going to let this Q&A go for a while. And you still have last statement on everything today.
Great.
All right,
I'll keep it in my pocket.
Thank you, doctors. That was very interesting and informative. I'd love to know how accurate those blood tests really are.
I mean, I can speak to that if you want. Well, we have metrics that are very technical. There's one metric that's very statistical called accuracy. And it tells you how sensitive the test is to the presence of the disease and how specific the result is to the presence of the disease. And then you combine that measure into a metric called accuracy, which is a statistical measure. And they're about 90% accurate. I mean, we want to see that kind of accuracy to have a practical blood test because we don't want to have a lot of false positives and false negatives. So they're pretty good, and they're going to get better, and they're about 85 to 90% accurate, which is, by industry standards, for any blood test, pretty acceptable.
So some people, you're saying they're negative, but they could be positive because the test isn't absolutely accurate?
Well, no test on the market is absolutely 100% accurate. So this is the kind of test that would be approved by the FDA, for example.
Thank you.
I'll just reinforce that we think of tests like your cholesterol level as being very definitive, but as a matter of fact, these tests all have false negatives and false positives. And I often use the example that about 10% of the time when your surgeon takes your appendix out, it's normal. So there's a lot of opportunities for misdiagnoses.
Well, we talked just before your entry up here, and I have to say, congratulate you. You did a really good job here, and then being in the business and so on. And of course, Dr. Jeff, I want to talk to you how we can get our product into that prevention trial, because that's is something that is very, very exciting. However, since we are in that field and we're working on longevity, aging, health span, and Alzheimer's, Parkinson's, and so on, how do you see the FDA side? Prevention is not really on the FDA radar screen. How can you get a trial that you say, hey, that now the product is approved for prevention?
Well, if you think about statins, they were initially approved for what's called secondary prevention. So what that means is take people that had a heart attack, right? So they had high cholesterol, they had a heart attack, and now you want to prevent them from having a second heart attack. So secondary prevention means people already have the disease, both pathologically and clinically, and then you want to prevent the disease from progressing. That's a treatment trial. So these would typically what we've done in Alzheimer's disease, have people that have mild cognitive impairment or early dementia, and we try to treat them to slow it down. Primary prevention, there's two ways to look at it. One is taking people that have very early or no symptoms, but you know that they have the disease in their brain, and try to prevent them from developing symptoms. Or taking people that have no pathology and trying to prevent them from having any pathology. That would be two forms of primary prevention. And all those kinds of clinical trials are going on now. And the FDA can approve any one of those indications. So they can approve a drug for the secondary prevention of heart disease, or they can approve a drug for the primary prevention of heart disease. In the case of statins, doctors, because of the way that the statins work and so on, the drug is really approved based on what's called a surrogate marker, where the FDA recognizes that cholesterol is a risk factor for heart disease per se. And so doctors in that context can prescribe anybody that has a high cholesterol, whether it's primary prevention or secondary prevention, they can prescribe a drug, statin, that lowers cholesterol because there's good relationship between a high cholesterol and heart disease of any kind. The same thing's going on in our field. The Biogen drug was really a drug for secondary prevention to slow it down. But there are also trials that they're doing for primary prevention that's being done with these monoclonal antibodies. So our field, instead of cholesterol, we're looking at amyloid as the cholesterol equivalent.
Yeah, I just thought that the difference is when you really look earlier, versus then when you really see like the Biogen with the malfunction impairment, that's the indication that they have drug approval and so on. So,
Yeah, you need a.
Let me just say while you're getting the microphone, I think it's going to be complex. You know, these are, the FDA wants to be convinced, and it takes a lot of data to convince them. One of the things they always ask is about clinical meaningfulness. How meaningful is your intervention? Which I think we all want to know, right? Before we spend money and invest as a society, we want to know that it's clinically meaningful. So how do we show clinical meaningfulness of treating a person who's already normal? So we just published a paper on this last week because we really want to struggle with this concept, and that we introduced two new ideas. One is predictive benefit based on biomarkers, and one is cumulative benefit based on prolonged exposure. And we think that both of those ideas should be part of the dialogue of clinical meaningfulness when you're treating people extremely early in the disease, because we want to treat people early in a disease. We want to keep our minds and our parents' minds as good as possible for as long as possible, not after they already have the disease. So we need to struggle with these, but I think it is going to be complicated.
And Jeff reminds me of one more thing to say. You think about the risks of developing a drug, and I spoke to that a little earlier, how high risk it is and how costly. That's to get FDA approval, right? Today, in the United States and all over the world, Europe, Australia, Japan, you name it, there's another hurdle, which is payer risk. In other words, is Medicare going to pay for that drug? Now, the regulatory standard for Medicare to pay for a drug are 3 words: reasonable and necessary, vague words. But we've just seen this play out with the Biogen drug, because they've decided that the Biogen drug may not be reasonable and necessary to pay for. And they're not supposed to evaluate costs. But what happened was that Medicare increased the Part B premiums in anticipation of the budget impact of that Biogen drug, even though they're not supposed to. And so Medicare is not paying for that drug in full, even though I think we'd agree there's some effect there. But what I'm saying is that as healthcare budgets become so expensive, and these drugs are so expensive to develop, there's this other hurdle. You get through all this science, 15 years of science, you prove that the drug works in phase 3. FDA even approves the drug, but you still don't know it's going to be paid for. Imagine you're an executive at some big pharmaceutical company, and you've got to decide whether you're going to put aside a budget of $2 or $3 billion to take a risk on a molecule that you can't be assured is going to be paid for, even if it works. That's the new society that we live in. The brain scan that we developed for the diagnosis of Alzheimer's disease is incredible. I mean, like I say, I can order that. You go down the block and get the scan, and the next morning I can tell you with about 90% certainty if you have Alzheimer's disease in your head. It's incredible. It's revolutionary. Medicare won't pay for it. And we could talk about why. So there's a payer risk.
One of the things you mentioned is technology, and there's a lot of things happening in the clinical trials and the whole thing. One is digitization, and the next one is artificial intelligence. You have to speed up the process. Bill Gates said it by about a year or more. Can you comment on those two things, and also comment on messenger RNA, if that is a part of the process? Those are my opening questions. Then I want you to comment more on sugar.
Do we have to remember all 3 questions?
Yeah, sugar.
I didn't quite hear the question.
Well, he wants to know about the role of AI in drug development.
Okay.
Can you do that?
It's so fascinating to see AI being applied so broadly in drug development now. Whether it is, as Howard was talking about, screening compounds very early, or looking at characteristics of compounds that correlate with clinical benefit, or looking at patient characteristics in electronic medical records. We use it now in our annual review of the drugs that are in the pipeline. It's fantastic. We can do in seconds what it used to take us, days to do by hand. So the application of AI is very broad, and what I see, and we were talking about this last night, that pharmaceutical companies are purchasing the AI companies that are coming along. So you can see that pharma sees that this is a powerful, powerful tool for exploring and supporting what they want to do.
I think I remember your third question, which was about diabetes drugs, if I'm not mistaken. And mRNA. Yeah. Well, I'm not sure about how mRNA will play out. I think it's very different to get a vaccine, to have the mRNA to make the COVID proteins that ultimately your body reacts to. I think it's a very different process to try to fix a complicated disease like Alzheimer's disease, which has multiple pathways. I think in genetic diseases where there's one mutation and there's a protein that's being made that's, let's say, abnormal or deficient in some way, that I could imagine a day when mRNA could be engineered to make the normal protein. And you know, if you have like 50% abnormal protein and 50% normal protein, then you might compensate and cure the disease or treat the disease. So I could imagine a day, and I know that that's going on now, that people are thinking about applications of mRNA for chronic diseases, you know, beyond its current application, which is obviously for vaccines and infections. I don't know if that makes sense, but that's my view of where it is now.
And I'd just say that there is one drug that addresses RNA. The tau protein that Howard mentioned is subject to post-translational RNA modifications. So if you could control the RNA, you could prevent the changes that lead to the neurofibrillary tangle. And so there is one trial that is trying to do exactly that. It's complicated because the drug has to be given in the spinal fluid. So you have to have a spinal tap, and the drug is injected into the spinal fluid around the spinal cord so it'll circulate up to the brain. But if we could, very often, if we can prove that a drug works in a complicated trial, we can then move that technology into a less complicated trial.
And then about diabetes, there's a drug on the market, the most widely used diabetes drug is Metformin. And we funded a study of Metformin for Alzheimer's disease several years ago at Columbia University, which showed some positive effects on glucose metabolism using a specialized kind of PET scan that measures glucose metabolism in the brain. The idea behind it is that even people without diabetes, as we age, the aging brain becomes insulin resistant. And the brain is completely very dependent on glucose. So if you think about a diabetic, any diabetic, if they take too much insulin and their blood sugar drops below 60, something like that, what's the first thing that happens to them? They go immediately unconscious, immediately. That's how sensitive the brain is to glucose. The brain is 3% of the body weight and uses 20 to 25% of the body's energy derived from either glucose or oxygen at any given moment. Now imagine that deficit on a chronic basis with a reduction in insulin sensitivity and insulin resistance in the brain. We know that exists with aging, so that the brain is less efficient in using glucose. That ultimately could lead to, and has been shown to lead to, neurodegeneration. And that's kind of the rationale of repurposing insulin sensitizers for Alzheimer's disease. So Dr. Luxinger, who did the trial at Columbia, just got about a $40 million grant to conduct a large trial of Metformin for Alzheimer's disease. And our foundation just funded a clinical trial, a prevention trial of Metformin in combination with lifestyle risk factor management and diabetes and hypertension management in Finland, which is the largest and most the most advanced prevention trial in the world. Dr. Kivipelto, who's a member of our board, conducted this randomized trial to prove that prevention really works, and that trial is being replicated now in 45 countries around the world. What we want to do is take the heart disease model. So if you want to prevent heart disease, you exercise, you take your diet, you sleep, you avoid stress, et cetera, and you add a statin. Well, we want to do the same thing in Alzheimer's. Exercise works. We know it works. Diet works. We know that it works. Sleep is good. et cetera, and we want to add metformin, which is actually the leading anti-aging drug in the whole field of aging. And there's a clinical trial being conducted now out of Albert Einstein in New York, funded by the NIH, which is the first time that FDA has even looked at an anti-aging drug that might come to market to prevent any disease of aging with one drug, and that drug is metformin. So it's not just through insulin, but metformin has other effects in the body that are anti-aging effects. So we're entering into a new era, and we just funded, we're honoring Miranda Orr out of Wake Forest University. She's developing a new class of drugs for Alzheimer's called senolytics. And this addresses a fundamental mechanism of aging cells, which is as cells age, they refuse to die. They stay around. There's normally, as cells get damaged, they automatically undergo a suicide pathway and they get out of the way. But with aging, and you get senescent cells that refuse to die, and they become very inflammatory. And these cells need to be eliminated. And so we're repurposing a cancer drug in a combination for the use of Alzheimer's disease to get rid of senescent cells in the brain, which we know are there. And this is the first of a new class of drugs called senolytics that address fundamental pathways of aging in the brain.
I want to add one thing to that, which is senolytics may not just have an effect on the brain, because these cells are accumulating in many organs in the body. So one of the ideas of using these drugs that address aspects of aging is that the brain would be one responsive organ, but so might your knees, so might your heart, so might your kidneys. So I think there's going to be a really interesting, very much wider set of outcomes in those trials than we've seen in any other trials.
And that's a really important point, because it goes back to you as a clinical trialist. How do you design a trial that could get an FDA indication that says it's anti-aging? And what they're doing is brilliant, because they're looking at all the diseases of aging, diabetes, hypertension, pancreatic disease, et cetera, et cetera, et cetera. And they're saying that if metformin is really an anti-aging drug, it should prevent not just brain disease, but all of the diseases. So the outcome measure for an anti-aging drug that could get an FDA indication is showing that metformin reduces the risk of onset of all of the diseases of aging and old age. And if that could happen, that would really be amazing.
Okay. In terms of lifestyle and diet and prevention, what do you think of the role of alcohol? Is it something, if you're concerned about Alzheimer's, you should avoid completely or use moderately?
Well, as a person who enjoys wine with dinner, I'll just say that I have a conflict of interest in my answer here. But the data are reasonable that, if anything, moderate alcohol use reduces the likelihood of getting Alzheimer's disease. And high alcohol use impairs cognition. It may not make Alzheimer worse, but it impairs cognition. So I think the challenge is moderate alcohol use. That may, there may be many aspects of this. Resveratrol is one of the components, and maybe long-term exposure to resveratrol is important. Maybe it has to do with stress reduction. Maybe it has to do with other aspects of diet that might go along with wine. There was an interesting observation that the first scientist to make the observation that wine was likely good for you was Jean-Marc Orgorgazot, who was located in Bordeaux. And I always thought Jean-Marc had a little bit of a conflict of interest in that study as well, but it has stood up over time.
Well, I'm going to make a little dissenting point of view here, au contraire. There was just a study that came out, so I won't hold you to it, which looked back at this epidemiological data that suggests that wine is reducing the risk of Alzheimer's. That's where we get it from, these epidemiological studies that only show an association, doesn't show a causality. So it's a big problem with epidemiological studies. And what they found was that the decreased risk of Alzheimer's disease and chronic diseases with the use of a few glasses of alcohol, of wine, let's say a day, was actually eliminated when the data were reanalyzed for socioeconomic status. So when they reanalyzed the data for socioeconomic status, the benefit of alcohol disappeared. But there's a practical point of view here, and I've come around to this. When I see a patient into my office who comes in, and this is really where being a physician, I think, is really helpful in helping us to make judgments and opinions about this stuff. Somebody comes into my office with memory problems. They're 75 years old, and I take a history. Do you drink alcohol? Yeah. How much do you drink? Well, I only drink 2 glasses a day, and it's only this much. And the wife is going, yeah. Oh, maybe it's 3 glasses, and maybe it's this much. Doesn't matter to me. Alcohol is a neurotoxin. Alcohol is a neurotoxin. You have decreased brain reserve as you age. You're less able to tolerate your liver function, your metabolism declines, your blood alcohol levels go up higher with the same amount of drink. And I'm sure that the brain is more susceptible to the neurotoxic effects of alcohol. And anyway, I want to optimize somebody's cognitive function. So I will just tell them, I think you should just, don't tell me you're going to go down to just 1 glass a day. I just want you to stop drinking alcohol and let's see what happens. And, sometimes they say, gee, doc, I've been off alcohol 3-4 weeks and I feel so much better. But either way, that's my clinical opinion. I want to protect, if it's a really quality of life issue for them, they say, I got to have that red wine every night. Okay, you're 75, you want to have the red wine, that's fine. But I would argue, I would recommend it. I do recommend against it. I think that we're trying to do everything to preserve what's left there. And I've come around to that kind of opinion.
I don't know if that's a great fundraising pitch, but anyway. I have 2 unrelated questions. One is, how does NLRP3 fit into this whole mosaic? And the second, how do you deal with the inherent conflicts of interest in venture philanthropy?
Well, I'll take the second question first. I'm not sure when you say the inherent conflicts of interest exactly what you mean.
Well, I mean, if you're actually looking at the potential returns of one investment versus another, I mean, you have potential conflicts of interest, right?
I'm not sure.
So,
Let me tell you
some
how.
foundations just don't do it for reasons of conflict.
Well,
I'm not saying right or wrong.
So there's IRS regulations around the ability of private foundations to invest in public or private companies, okay? And the way the IRS regulates that is that the foundation has to show a clear purpose in making the investment, that the investment is clearly for the purposes of advancing the charity's mission. Ford Foundation pioneered this in terms of conflicts of interest where, what happened was in the 1960s there were contractors who, did concrete building of apartment houses, and the contractor set up a private family foundation, used tax-exempt dollars to give grants to the for-profit entity that was building low-income housing for poor people. And so there was a self-inurement there. So for private foundations, there's an issue of self-inurement. For public charities, we have freedom to operate. Our primary purpose in making investments has to be compatible with our mission. Our mission is to accelerate drug development. Drug development is a commercial interest. And the IRS knows what we're doing because when we incorporated with the IRS, we told them that we were going to be making investments. And now it's much more prevalent than it was in 1998. The other conflict of interest is sort of insider information, let's say. And we have a very strict conflict of interest policy with our boards about how the boards can make investments if they want to, which actually happens almost never. But I think the issue around whether this is a legitimate, legal kind of process, venture philanthropy, I would say the answer is absolutely yes. This is something that's come of age. And I think in terms of the sectors of our economy and to address diseases, it's become very important that big foundations like Juvenile Diabetes Research Foundation Cystic Fibrosis Foundation, they're all doing this kind of thing now because it's become widespread, because of the recognition that this plays an important role in taking risk that for-profit investors don't want to take. That's our purpose. Okay, so we have a role in the investment ecosystem. Your other question was about
Let me just comment on that, because I think this is a really important question, right? Because the public perception of philanthropy is so important. And I think there are two things about ADDF that are helpful. One is the Scientific Advisory Board reviews everything with great due diligence and then makes a recommendation. And that recommendation, I think, is, if it's a consensus recommendation, is usually followed. So it's not like a person is making a decision. The other is that the salaries of the ADDF personnel do not depend on the success of the investment. So this it's independent, and it's not like they would sort of funnel money one way in order to get personal enrichment out of that. But I think the question is so important because we want clear transparency about that.
Yeah,
So the other question is about Parkinson's disease.
NLRP3.
Sorry.
I think you asked about NLRP3.
Oh, is it? Oh, so this is one of the inflammatory pathways, Denali has one set of drugs. That's a pharmaceutical company that is developing one set of drugs addressing that target. There may be others. Do you have one of those drugs in your pipeline?
Well, we've seen university programs on NLRP3 also, and I think it's an important target. But, understanding the biology is one thing, and making a target druggable and getting drugs is a totally different thing. And I think NLRP3 has been difficult to drug, basically. It's intracellular, it's intranuclear, so you have to have a special kind of drug that can get through the blood-brain barrier, get through the cell membrane, get through the nuclear membrane, and affect NLRP3 within the nucleus of the cell. So it's been it's been an obvious target, but a hard target to drug. That would be my more global opinion. So. And by the way, we often, just to get back to your point about venture philanthropy, we have an investment committee that's steered by our board. They're very careful about what we do. They want to know the scientific justification. Yeah, okay. And we a lot of times for-profit investors will come to us for validation, and you'll hear it from biotech companies. If ADDF validates it, it's an independent voice that is often very valuable to small companies.
We have time for one more question if we have some right here, because we have somebody who had it, and they're going to be around, so don't worry. Go ahead.
Are the researchers for Alzheimer's doing any other, looking into any other treatments besides drugs like stem cell or oxygen therapy?
So I can take part of that, and the answer is absolutely yes. There's quite a few novel therapies. I think there are 6 stem cell therapies very early, phase 1, when they're just starting to see how they're going to work. There's 1 at phase 2. I don't think there's an oxygen therapy trial in the pipeline. There are some gene therapies that are starting to be looked at. One particularly important is funded by ADDF. So we're seeing non-drugs come into the pipeline, cells and genes. And then Howard was making the point about the importance of exercise and diet folded in with a drug trial. And we think there are maybe ways of amplifying drugs that we haven't taken advantage of yet. And that's starting to come into the pipeline as well.
Good.
So a couple of things. One, Jeff, you put something in your pocket earlier that you were going to have a thought that you were going to say to Howard before we started Q&A. Do you remember?
So Howard was talking about digital biomarkers. And one of the companies that I've been consulting with is called NQ. And NQ has a way of measuring your speed of response on your computer or on your cell phone when you put this device on, which of course you have to give permission for. And it turns out to be remarkably sensitive for Parkinson's disease. We've shown that, and it looks like it will be sensitive to early changes in Alzheimer's as well. So that's a completely passive measure. Don't have to do anything. We just collect the data, and we do it remotely, and we could follow somebody's treatment that way, for example. We could follow whether people are changing that way. We think that that's the kind of thing that we're going to see emerging in terms of digital technologies.
Well, and the advantage of that is, in most clinical trials today, the patients come in every 2 months, every 3 months, maybe once a month, and depending on how they feel that day, cognitive testing can be very variable. Whereas with digital testing, you can get data points every day. You can get data points 5 times a day. And that is going to really improve the way we do trials and the accuracy of trials, the efficiency of trials also, because we'll have so much more data on a longitudinal basis.
And that comes back to the AI question, because of course this amount of data can only be analyzed by very powerful analytic tools.
First of all, can we have a round of applause? How great were they?
Really great question.
That's it. I want to do a couple of things here just to wrap up. This is the end of our scientific series for the season. Heidi and Tom, again, thank you for bringing this to Palm Beach. You are fabulous. Any of you who have been here, or even for tonight, there's a couple of clear statements that we can make because we hear about a lot of excitement here. One, there are tests here now, and they're only going to get better going forward. If you want to know, you can know. Two, if you want to know, even if you don't want to know, there are things you can do right now, starting tonight, today, to help prevent or push out, scientifically proven. Three, you heard about these repurposed drugs, these individual molecule drugs, new treatments. This is coming now, and you heard particularly because of what we're able to do with technology, biomarkers. The speed of these is going to come quicker. So 2 definites. If you want to know, you can know, and the tests are only going to get better in the next 3 to 5 years. You can prevent and do things to push out right here, right now, and today. And 3, there are a variety of treatments that we're going to hear much quicker, next 3 to 5 to 7 to 10 years, pass or fail on them. And what it's going to be, all of us here, I hate to say, as we're here, all of us are aging. We all age differently. We all have different biology and makeup. So when Howard talked earlier, it will be combination therapies, precision medicine. These opportunities I will see in our lifetime. The generation immediately behind us, these are going to be prescriptions. And the generation behind that, it's going to be stay on the protocol. You may forget where your keys are, but you're always going to know what your keys are for. And you're going to pass from something else, okay? But I say this because what you have helped fund is speeding more of those diagnostic tools, tests, more of the prevention science, and most importantly, these clinical trials to figure out you different than me, different than you, and what's the precision we need to put into that. So, yay, as Jeff would say. Also, we have refreshments. The 2 docs are going to be here for at least a half hour, so have at them. Thank you.